Hi, everyone. I would like to thank everyone for attending Life Technologies Multicolor Flow Panel Design webinar today. It is my great privilege to introduce Dr. Holden Maker, the Director of the Human Immune Monitoring Center at Stanford University. His research focuses on cellular immune responses to chronic pathogens in cancer and the correlation of those responses with immune protection. Dr. Maker's lab also routinely runs flow cytometry assays, and his lab is routinely running 8 to 10 color panels. So I consider him an expert in this multicolor panel design. So thanks, Holden, for being willing to host our webinar today.
Thanks, Gail, and appreciate Life Technologies for hosting and everyone for attending this webinar. I hope we can keep it stimulating enough. I know it's sort of difficult to look at a screen and not see a person and not see a pointer waving around, but we'll do our best. So this is a topic which gets a lot of attention because I think there are newcomers to the field who sort of consider a flow cytometer as they would a power strip where you can plug in 4 or 6 or 8 different cords and you don't have to worry about which one you plug where. Unfortunately, a flow cytometer doesn't work that way, and there are differences in the fluorochromes, in the cytometers themselves and their capabilities, and so the design of panels in flow cytometry has become a little bit of an art, and that's the art that I'm going to hopefully share with you today. So the sequence of things we're going to talk about begin with your instrument. And so point number 1 is really about knowing your instrument and what it can do. And then we're going to get into the reagent side in terms of picking antibodies and picking fluorochromes to go with them. From there, I want to talk a little bit about controls and standardization, which I consider a part of your panel design because controls are potentially adding extra tubes to your panel. And standardization is something you want to think about if you're going to do that panel more than once. And most of the time, if you take the trouble to build these multicolor panels, you're going to want to use them over a period of time and compare results. And then finally, I'm going to talk a little bit at the end about mass cytometry and what the issues of panel design are there. And for those of you who aren't familiar with that platform, I will introduce it briefly. All right, so let's start with the instrument. And many of you will probably be using an instrument that's in a core facility, and so you may not actually have the opportunity to do things like change out filter sets. It may be all pre-configured. But that's okay. What you should know is whether you have the ability to run certain fluorochromes, which ones are brighter, and which ones are essentially the best suited to that particular instrument. And hopefully, if you are using a core facility, they can provide you some information on that. They should be able to tell you, these are the channels. We have a blue laser, we have a red laser, we have a violet laser, and you can do these colors off of the blue, these off of the red, and these off of the violet, for example. And so the rules here then become, of those choices, choose the brightest fluorochromes, okay? But at the same time, minimize the spillover between them. And so I'll give you an example. If you are doing an experiment of, let's say, only a few colors, and you might as well, and let's say your machine has the ability to do 8 colors, you might as well pick the brightest few of those to use for your experiment. Whereas if you're going to do 8 colors, and your experiment can do your instrument can do 8 colors, then you have less leeway. But if you have the choice, pick the brightest fluorochromes. But there are some fluorochromes that are very bright but very difficult to use together. And an example there would be, for example, PE Sci5 and APC. They have notorious spillover between them, And for the most part, you want to avoid using them together unless they're marking completely independent populations of cells. And I'll get to an example like that in a little bit later in the talk. So those are the two rules about your instrument and which fluorochromes and which channels you want to take advantage of when you're doing a particular experiment. Now, I've used the word bright already, and this is a word we throw around a lot, but we should define it. What do we mean by bright? Well, in this case, I should start by saying that we're talking about the end result of staining with a particular antibody conjugate. I'm not talking about the quantum fluorescence of an individual fluorochrome molecule. I'm talking about the net result. When you stain with an antibody, how much resolution do you have between your positive peak and your negative peak? And so that's why I call it resolution sensitivity. Okay, so that's the distance there between the red and the blue peak. But there's another factor, which is the spread of those peaks. And in particular, with multicolor flow, it's the negative peak that often spreads because of spillover from other channels, something that we're going to talk about in a bit more detail. And so taking some measure of the width of that negative peak is also important for resolution sensitivity. And so we have this formula that was first put forth by Dave Parks, and it's now a very generally accepted kind of calculation. There's a little bit different versions of it, but it's essentially called a stain index, and it is the difference between the positive and the negative peak medians divided by some measure of the width of the negative peak, and we like to use 2 times the robust standard deviation, which is a statistic that you can get from your software. All right, so that's what we mean by good resolution sensitivity, and that's what we in turn mean by brightness. Now, having said that, you will probably ask, okay, so how bright are various different fluorochromes that I like to use and that my machine can do? Well, here's a set of fluorochromes, some of the most common ones that you will encounter in the multicolor world, and the associated stain index done with a CD4 antibody, the same clone actually of CD4, conjugated to each of these fluorochromes. And I have to put another caveat on there. This was done on one particular instrument. It happened to be a BD LSR2. But the stain index is clearly related to both the antibody that you used and the instrument and its configuration. So that's why you have these numbers, and they should be taken as relative numbers for a particular instrument. Another instrument might be a little bit different. So you can see that PE Sci5, PE, APC are really at the top of the list. Alexa Fluor 647 is not far behind, and this constitutes essentially our group of the brightest fluorochromes. There's a couple more good ones in the middle, and then we get down to our dimmer ones, which are not going to give you as much resolution sensitivity, and I will tell you a little bit later how that's going to affect your choice of what antibodies to put on. All right, so I talked already a little bit about spillover, and I want to make the point now that spillover really does affect your resolution sensitivity. So the stain indexes that you saw on the last slide are predicated on using just that one single antibody by itself with nothing in any of the other channels. But in the real world, in multicolor flow, we use multiple antibodies together, so we have spillover, and that affects the resolution sensitivity. Here you can see that in the boxes, there are filters designed to capture the fluorescence of both FITC and PE, and to capture them pretty much separately. But as you see in the purple balloon there, the FITC contributes a fair amount of extra fluorescence to the PE filter, within the PE filter, within the bounds of that bandpass. And so you're going to have more spillover of FITC into PE than the other way around. Because if you look at the yellow peak, it only very little bit overlaps with the green filter for FITC. And by the way, this is a nice web tool that you can use to look at these sorts of things, and they're available from multiple suppliers. This one happens to be the Life Technologies web tool for picking filter sets and looking at fluorochromes and spillovers. All right, so I'm also now here showing in turquoise the 488 nanometer blue laser line, which excites both of these fluorochromes, FITC and PE. And so a little bonus question for all of you is, why is the FITC filter in the green rectangle so narrow when the FITC emission peak, which is the curved green line, is relatively broad? We could capture more of it, right? We could use a broader filter. Why does that filter not extend down lower to capture more of the FITC peak? Well, I'll answer it for you, but maybe you've already figured it out. It's because of that blue laser line. You can't get too close to it, or else you're going to get a lot of the laser light going directly into the filter, and that would be bad. That would hugely increase your background. Okay, so that's a little bit about spillover, and I'll get to compensation and so on in a moment when we are actually talking about choosing antibodies for different fluorochromes. But right now we're still sort of talking about basics of the instrument and the choice of fluorochromes. And so I want to wrap up this section by telling you what are some sort of typical multicolor options with different lasers. And you will encounter others out there. There are newer machines that are using all kinds of different excitation sources, but a rather traditional setup from a number of vendors would have lasers that look something like this, maybe 3 of these, maybe all 4, a blue, a red, a violet, and possibly a UV laser. And these are just some of the most common fluorochromes and best fluorochromes that you can use on these different channels. I've colored them a bit, and where you see, for example, the red writing, like PE Texas Red and Alexa Fluor 700 and so on, those would be my second-tier choices for those laser lines. So in other words, if we look at the red laser, I would prefer to use APC or Alexa Fluor 647, and then APC H7 or APC Alexa Fluor 750 before I turn to Alexa Fluor 700. And that's because Alexa Fluor 700 is dimmer, and it sits between the other two in its emission, so it's going to introduce a bit more spillover. So the same goes for the violet laser. You can see I put one kind of second-tier choice there. Same for the blue laser. There's a second-tier choice. But basically, you can fill up all of these, and if you count them up, it's somewhere around 14 colors. And I think with the new dyes that we have now, especially all of these brilliant violet dyes and Q dots that are available for violet laser excitation, you can actually get up to 12 to 14 colors without a lot of problems. And that has not been true for very long because we didn't have that many good fluorochrome choices. All right. So moving on to the second part, which is highly related, we're going to now match our antibody specificities with the fluorochromes that we've chosen. We've chosen the fluorochromes based on our instrument, its capabilities, and what are the bright fluorochromes that are available for that instrument that have relatively little spillover between them. So now we say, all right, with that list of fluorochromes, let's pick where to put our various antibody specificity. And the rules are basically these, and you will have probably heard these before in some version or another. They basically are put your bright antibodies on your dim fluorochromes, and vice versa. Reserve the brightest fluorochromes for your antibodies that have either very low abundance targets or are for some other reason very dim. And number 2, avoid spillover from the bright cell populations into the detectors that require high sensitivity. And I'm going to have to give you an example to really explain what I mean by that, but it gets at that point that I already made about spillover affecting the resolution sensitivity. And then there are some special issues that apply to tandems, mostly to the size 7 tandems, and so we're going to talk just a little bit about those. They have some propensity to break down and cause extra spillover in their parental channel, and there are some ways to help avoid that or deal with it. All right, so getting back to this spillover issue, here you see in the left panel an uncompensated set of FITC beads being run on an instrument and looking at both FITC and in the y-axis PE fluorescence. And you see this blue population kind of up at a diagonal, right? It's definitely staining for FITC, but it also has PE fluorescence, even though there's no PE in these beads. So that's because of spillover. What do we do about it? We apply compensation. We measure from these single color compensation controls. We measure the amount of fluorescence in both FITC and PE, and we essentially apply a matrix that subtracts out the PE signal on average. And so you end up with the result on the right, which is compensated, and now you see that the blue population lines up in the y dimension with the green population. There is no net PE fluorescence to the blue population anymore after compensation. But what happened? Look at the spread of that blue population compared to how tight it was in the uncompensated condition. Well, that's actually kind of a visual artifact because this is a log scale. And so when you move something from high up on the log scale to lower on the log scale, it spreads out. The variance of the population hasn't really changed, but now it appears to spread further. And because of the spillover into the PE channel, there is now less ability to resolve a very dim signal that might sit right at the top of that blue streak in the PE dimension than there would be if there had been no PE fluorescence in the uncompensated condition. I've got a reference there at the bottom. You can read a bit more about this kind of thing, but that's the basic idea. The more spillover you have from an adjacent channel, the harder it is to resolve a dim signal in that channel. In this instance, it's the PE. Okay, that would be harder to resolve. Now, it's still a very bright fluorochrome, and we can certainly use FITC and PE together. That's not an issue, but the phenomenon is there, and we call it data spread. All right, here's a different pair of fluorochromes, but a real-life example of how that resolution sensitivity can become degraded. In this case, we used a fluorochrome called AmCyan, which is actually not a very good one. It's kind of fallen out of fashion now as there are better alternatives. But AmCyan has a propensity to spill over into FITC. And so what you see, what I've done here in this experiment, which is a contrived example, is I've used the brightest thing that I can find in AmCyan, which is CD45. And in the FITC channel, I've chosen something that's just a little less bright, which is CD19. And you can see that without the CD45 AmCyan, the CD19 resolution in the left panel is beautiful. It's very nice. But when we add in the CD45 AmCyan to the cocktail, now look, the two peaks are almost on top of each other. There's still a little bit of resolution, but it's pretty marginal. Why? Because of that spread of the negative population from the spillover of AmCyan into FITC. The negative population now, it's not only shifted to the right, it's actually spread, and if we didn't have a log scale but a bi-exponential scale, you'd see this, that it actually spreads way below zero, and that now means that it's hard to resolve those CD19 positive cells. But there's a very important caveat to this, and that's at the bottom. It's only an issue when the two markers, CD45 and CD19, are co-expressed on the same population. If I had chosen CD3, in my AMP-cyan channel versus CD19, that's a T cell marker versus a B cell marker, this really wouldn't be an issue because I could gate out all the CD3 positive cells before I even look at the CD19 staining, and therefore I could sort of bypass this whole issue. So, a very important rule about spillover is that when you are forced to use two channels that have a lot of spillover between them, try to use them for mutually exclusive cell populations, and then you won't have much problem. All right. Getting to the issue of the tandem dyes, again, this is mostly applying to Cy7 dyes, but the first point actually applies to pretty much all tandems. And I should maybe give a quick introduction to what a tandem dye is for those who aren't familiar. This is where you have two fluorochromes basically in combination, and they work by the first fluorochrome absorbing the light and fluorescing it and passing that energy on to the second fluorochrome, which then emits at a longer wavelength. So it's a way that we can get more fluorochromes into a particular laser line because we can excite them all at the same point, but we can get longer wavelength emissions by making some tandems. And so examples of tandems are PE Cy5, APC H7, PerCP Cy5.5. All of these are tandem dyes. And the first point applies to most all of the tandems. Their compensation requirements can vary between different lots of the same antibody, and even the same antibody when stored over time can change a bit. Now, that still is not equally true of all tandems, and the Cy7 tandems tend to be the worst. But the bottom line is that if you're using these tandems, you probably should do your compensation experiment by experiment, or else you may see some drift over time. And it's a good practice to do that anyway. So that's the first point. The second point is that the Cy7 tandems in particular degrade with light, temperature, and fixation. So you really want to minimize your exposure to these conditions. And there are some solutions out there. There's a stabilizing fixative that's available that helps to keep that degradation to a minimum. So those are the caveats of the tandem dyes. And on the next slide, I'm going to show you an example of kind of the worst case scenario of what happens if you don't pay attention to these rules. All right, so this is an experiment which we actually did back when we were first getting into 8-color flow cytometry. And we thought this was going to be a good idea. We were going to take advantage of these new PE Cy7 and APC Cy7 tandems, use them for CD4 and CD8, and we were going to put our cytokine antibodies in the PE and the APC channels, which are the parent dyes of those tandems. And I say, I'm admitting we thought it was a good idea at the time, but it clearly isn't. Because what's happening, if you look at the top row in A, when we have CD4 in APC Cy7, and we have CD8 in PE Cy7, that's the green population, these can degrade. And in the worst case, the way they start to degrade is to show up as false positive signals. There's a subpopulation there of events that you see in the middle and in the right-hand panel. They're circled, and they're beginning to look positive for interferon gamma APC or for IL-2 PE. And you might be fooled and think that these are in fact cytokine positive cells, but they're not. These samples weren't even incubated in Brufeldin A to accumulate intracellular cytokine. They can't be real positives, and we can prove that they're not real positives in B by taking out one of those parental dyes. We take out the CD4, which is the APC Cy7 in blue, and now we see that APC population for interferon gamma is completely gone. That's called an FMO control, fluorescence -1. We leave out one reagent and we see the effect on the cocktail. And here it clearly proves that that signal in APC is a false positive. And we could do the same thing for the PE, do an FMO for PE and show that that green false positive population goes away. So that's the worst case scenario, and because of the choices we made, trying to look at small populations that would be cytokine positive in the parental channels of these tandem dyes that we were using for bright and universal kinds of antigens like CD4 and CD8 was just not a good idea. So we rearranged our panel and put our cytokine antibodies elsewhere, our CD4 and CD8 not on these tandems. All right. Now, I say that's the worst case scenario, and in part that's because manufacturers have largely moved away from APC size 7 and replaced it with more stable alternatives like APC H7. And this is an experiment showing you the spillover of APC H7 reagents in the colored lines versus APC size 7s in the dotted black lines. And you can see that over time with light exposure, the size 7s degrade, and the APC H7s much less so. And I should mention that this is in the presence of that stabilizing fixative that I mentioned, so the degradation is actually quite slow. It takes a while, but you can get them to degrade even in the stabilizing fixative. All right. Now let's talk a little bit about controls and standardization. And again, controls are important because they are part of your panel. They are the things that you use to either set up your instrument for that panel or to gate that particular panel, or as a kind of biological control for the results of that panel. And so I've broken the controls into these three groups. The instrument setup controls are things that you use to either set your PMT voltages or your compensation. The gating controls are things that you use to set your positive-negative boundaries for different populations. They could be isotype controls. They could be these FMO controls. And the biological controls are the things that you use to compare, like an unstimulated versus a stimulated sample, or a healthy donor as a control for a diseased sample. They're the kinds of things that help with quality control of batches that you run. Okay, so I'm not going to dwell here about the instrument setup controls because that's really another topic, how to set your instrument optimally. You want to essentially achieve PMT voltages that minimize the amount of electronic noise contributing to your signals, and you want to be in a range where you can get the maximum resolution sensitivity for your particular fluorochromes without having your positive signals go off scale. That's the basics of PMT voltage setting, but we could talk about that in much greater detail, and we won't do that today. The compensation, as I alluded to, is best done per experiment, particularly if you're doing tandem dyes, and is mainly done with single color controls. That's the usual way. And there are algorithms now in all of the major manufacturers' softwares that will allow you to automatically calculate the compensation. It is something that you should check even after it's calculated for you by simply looking at your fully stained samples and making sure that the populations in each color combination that you can devise are looking appropriate, so that things are orthogonal to the axes when they should be, and there aren't some kind of diagonal streaks coming up that you would not expect. So that's something you should check, and that's all I'm really going to say about instrument setup. I want to dwell a little bit on the gating and biological controls here in the next slide with a specific example. So this is, again, an intracellular cytokine assay. You can tell that that's the kind of assay I like to do. And here we're looking at IL-2 on the x-axis in PE, and we have CD4 and FITC on the y-axis. And you can see in the very top our positive control, which is SEB stimulation. So there's lots of signal for IL-2 from both the CD4 positive and the CD4 negative cells. It's unfortunately a smear, and so it's a little bit hard to know where to set that boundary between positive and negative. But the way that I've drawn the quadrants in this particular example is based actually on the unstimulated control, which is the lower left. I've placed that y-axis boundary, the vertical boundary between positive and negative just beyond the edge of the unstimulated negative population. Okay? And then when you look at SEB, it seems to make sense. That seems to go pretty well. But there are other kinds of controls. You could use an FMO control, and some people have tried this to set your positive-negative boundary. But look how much further away that is. If you were looking at the FMO control only, you would have set that vertical boundary probably somewhat closer to the y-axis. And that would have cut into your true negative population when you looked at SEB or other experimental samples. And on the other hand, if you look at the isotype control, in this case, it's shifted much further to the right. And so if you were setting that vertical boundary based on the isotype control, you would have put it quite a bit further to the right, and then you would have missed some true positives in the test samples. So the point here is that you have a choice of controls, and some are better for certain purposes than others. In this case, a biological control, the unstimulated sample of the same donor and stained with all the same antibodies, is really the best gating control. But there are other cases where you don't have that kind of a choice, and maybe an FMO control is the best you can do. Rarely is an isotype control the best thing that you can do, because isotype antibodies can have this differential amount of background. And I would say if you choose an isotype control, you should try to match it to the background of your antibody, your test antibody. And you should use it in the context of all of the other antibodies in your cocktail, because just on its own, it doesn't account for the spillover from the other populations. All right. So I probably don't need to say anything more about that, but just in case some of you are still devoted fans of isotype controls, I will make the point with one more sentence, which is to say that this old concept of doing individual tubes with one isotype-specific antibody as a control is really pretty worthless. Or what I've seen some people do is throw in all of their isotype controls into one tube. So they have a IgG1 FITC, an IgG1 PE, an IgG2A per CP, and that's their control. That really isn't very helpful in the multicolor world, because you definitely can have different levels of background between isotype controls and test antibodies, and you definitely do not account for spillovers when you're doing something like all isotype control antibodies in one tube. All right, enough said about that. Let's go on to the point about standardization, which goes kind of hand in hand with controls. I think when you're designing your multicolor panels, you want to think about the long term. You want a panel to be reproducible over time. And the main point that I want to make here is that there are multiple ways to achieve this, but one which you might not have thought of, and therefore I threw in this slide, is to use lyophilized reagents. Lyophilization can provide increased stability so that you could create a bunch of cocktails that are lyophilized and keep them even at room temperature for a long time. We've used these things out to 2 years and more. And that way you can have one batch of reagents that last you for an entire study. It can be done from a couple of different vendors now as pre-configured plates. that would avoid errors of reagent addition, so you can have your cocktails laid out in a plate, as many wells as you want, and now you just add cells, and you don't have to worry about individually adding each particular antibody. That makes your complex experiments, where you have maybe multiple different stimulations and multiple different staining cocktails, much easier. And finally, it provides you, you can provide controls on those plates that are actually lyophilized cells to have some kind of run-to-run standardization, or you can have beads and other kinds of things for that purpose. So not something to be done in all cases, because there is usually a premium of cost associated with this, and it's really only practical if you're doing large studies, but something to keep in mind that actually does help with standardization. The other things that help with standardization are the things I already mentioned, including keeping some kind of batch controls in every batch that you run, whether that's a particular donor that you've frozen away in many vials, or whether it's some other kind of control that you include with every batch of samples, and then set some kind of QC criteria around it. But of course, one of the biggest ways that you can standardize is simply by designing a robust panel that doesn't have too many spillover problems, and therefore works most of the time without a lot of variation. Okay, now for the last few slides, I'm going to just move into something a little more fun, which is the application of panel design to mass cytometry. So for those of you who are unfamiliar, what is mass cytometry? Also known as CyTOF, because this is the instrument that is out there in the field to do this particular kind of technology. It's basically the use of a mass spectrometer as a readout for flow cytometry. So you're looking at individual cells, but they're stained with antibodies that have heavy metal ion tags on them rather than fluorescent tags. And so therefore you read them out on the mass spec, and this allows you to use many more antibodies in combination with essentially no spillover. And so if you look at these emission spectra of the fluorescence world, I'm showing about 8 different dyes there and how much overlap there is between their emission peaks. And you compare that to the right panel in mass cytometry, where you're looking at single isotopes, they are much more discretely read out on the instrument. So you can have lots more antibodies used together and essentially not have to worry about compensating. So I'm going to address a couple of points that relate to panel design in this world of mass cytometry. Even though on the surface it looks much, much simpler because you don't have the spillover issues to worry about. But I'm going to address the question of whether the choice of mass channel matters and whether there really is any spillover to worry about. And you probably are guessing at the answers to these questions, otherwise I wouldn't be spending time talking about them. But basically there are some differences across the mass window in sensitivity. And so this is an experiment that Mike Leipold in our group did a while back, in which he basically did solutions of individual heavy metal ions across the range of the CyTOF and looked at the counts that he was getting from equimolar solutions of each of those ions. And you can see there's a bit of jaggedness here, which I'm sure is just variation in the dilution that he made. But there's a kind of a general trend to peak somewhere up in the high 160s or close to 170 atomic mass. And it kind of plateaus or maybe goes down just a little bit above that. And your lowest atomic mass elements around the 140s are your least sensitive channels. But really this is a pretty shallow curve, and there's only maybe a 3-fold difference from the least sensitive to the most sensitive channel. which, if you recall the stain indexes that I showed you in one of the very early slides, there is a much bigger difference between the brightest and the dimmest fluorochromes in the fluorescence world. So it's better. It's not as big of a deal, but we do still reserve our most sensitive mass channels for our dimmest antibodies. Now what about spillover? Another experiment that Mike did is to look at the single isotope samples and read out how much of their signal was showing up in each of the different CyTOF channels. And so he's color-coded this, where red is highest and green is lowest. And along the diagonal, where you see that major kind of red streak going from the upper left to the lower right, that's the signal of the primary channel. That's what we want to see. We want to see almost all of the signal in that primary channel. But you'll notice a couple of things. So scattered about here and there are some yellowish or even reddish boxes, which generally are known isotopic contaminants of the primary channel. You'll also see some yellowish and reddish boxes right around that diagonal of that primary channel, oftentimes in the next highest mass channel, so what we call mass plus 1 or M plus 1 spillover. And that is a certain amount of detector infidelity where it's not making quite the right call on the isotope mass every single time. I'm told that that's actually much improved in the newest instrument, the CyTOF 2. And then there's yet a third observation here, which if you look off to the right, there's a kind of a second streak diagonally that mirrors the primary channel. And it's reddest, it's most noticeable at the lower atomic masses. And that is what we call oxidation. That's the incomplete ionization of these heavy metals, whereby they get an oxygen atom attached to them. And so now they show up in the mass plus 16 channel. That's something that you can regulate to a certain degree by tuning the instrument properly in terms of the makeup gas flow rate. But it is an issue that you have to think about. So for some of these lower atomic mass elements, you want to look at that plus 16 channel and think about what you're going to put there in terms of a specificity, because you will see a certain amount of spillover. Now, having said all that, none of these spillovers have ever driven us to the point where we've wanted to apply a compensation matrix to our data. The one exception that I can say for that was a case where we saw significant spillover by just a bad choice of specificities that we put on two different channels. And rather than try to sort it out by compensation, which really did not work very well, we simply switched the panel around. Okay? So the bottom line is that in mass cytometry, unlike fluorescence, you can essentially solve your problems with careful choice of where you put different reagents, and you don't really have to apply any compensation. I don't know anyone who actually does do compensation on the CyTOF. So, in conclusion, I would say the CyTOF, first of all, is a wonderful new platform. It's something that not everybody will have a need for or have access to in the immediate future, but if you do, it's a wonderful tool, but remember, it's not the solution to everything, and there are still some of these considerations of panel design that apply in terms of choices of where to place your different specificities. But overall, in conclusion, I want to say that, polychromatic flow is really easier than ever. We now have some really bright, nice choices of fluorochromes that can make combinations up to 12 or even 14 or 15 colors quite possible and not that difficult. But you do need to think about these points, which is basically that you select your fluorochromes for brightness and least spillovers, and that you optimize your choices of antibodies in terms of reagent brightness and the data spread between different channels. And then also think about your longitudinal QC. So there's a couple of references here that might be of interest. These were kind of tutorials that we wrote for Cytometry quite a while ago, but they're still pretty much applicable. There would be updates to them if we did them today, but there's still a start. And I just want to finish by acknowledging people that contributed data to this presentation, which include on the left side a big group of people from BD Biosciences who worked with me when I was there some time back, and on the right side some folks from Stanford that have contributed some of the recent data. Everything I've been talking about here is really research use only. These are not clinical applications, but I appreciate your attention, and I know it's been a lot of listening. So what I want to do with the last 10 minutes or so, if it takes that long, or we can take less time, but is I want to address questions. I see a couple of, or actually really only one person has put in a question so far, but if you'd like to, as we start this section, go ahead and write in any questions that you can have on this subject, and we can try to address them in the next few minutes. All right, one question that I see up here already is to which log value do you recommend to set the negative population when you adjust PMTs? So, I said, well, that's a subject kind of for another webinar, but the answer is that there isn't one place where you put the negative population in every channel. And that's because there are different levels of sensitivity of different PMTs, and every PMT is going to require a slightly different voltage to be optimized. So in some sense, we recommend you not to look at where the negative population lands. We rather would say you need to make a measurement of your sensitivity, which can be done in an automated way in some of the softwares, like the BD CS&T system, or you can do it manually by looking at beads and looking at the spread of a dim bead population and sort of picking the point where that spread becomes reduced and essentially plateaus at the lowest level. And then once you're there, you can go higher, but it doesn't really help. And so that's really where you want to base your fluorescence value, or your voltage values for your PMTs. And then your second consideration is where your positive peak lands. You don't want it to be off scale or out of the linear range. So that's a short answer to that question. Somebody else asked about the spectral analyzer from Sony. I didn't want to make sort of too many specific comments about particular instruments that are out there, but since the question is up, I'll just mention that there is a different way of analyzing multicolor data, which Sony has been embracing now with their new instrument, that essentially deconvolutes the signal of the entire spectrum as opposed to analyzing it through specific bandpass filters. And it's a much more complicated way of doing the analysis, so it requires a bit more computer power, but it gives you the chance to potentially better resolve signals that have spillover by looking at the entire spectrum and deconvolving the component parts. So that's the theory behind that spectral analyzer. All right. There's another question here. Do we have to use the 3 types of controls with each experiment? Unstained, isotype, FMO? The answer is no. Those are general types of controls, but which one you choose for a particular experiment depends on the experiment. And so I'll give an example with FMO controls. We really don't use FMO controls all that often in our routine panels. There's a couple of points where we use them. We use them when we're setting up a new panel and we want to measure what is the effect of leaving out each individual antibody. We'll at least do it for several of the critical ones in a new panel to see where is the spillover really, affecting things. So that's one use of the FMO. In terms of using it day to day in a panel that's already optimized, the only reason to do that is if you don't have another good way to gate between the positive and negative populations. Let's say that it's a smear, and let's say that even in a healthy control subject, there's some level of positivity. There's no cell population that's a clean negative. Well, then you probably want to use an FMO to be able to give you at least a consistent point at which to gate between positive and negative. But in other cases where the populations are resolved, why do an FMO? You don't need to. And the same goes for other kinds of controls. Unstained cells are always a good thing to run once in your experiment just to see where they lie and how much background there is with your stained samples. But really, no, you don't need to use every kind of control in any given experiment. Okay, looks like there's a little bit of conversation here around compensation. I'm going to just mention that compensation values are entirely dependent on the voltage setting that you're at relative between the two detectors in question. And so a compensation value of 80% or even 100% is not very indicative of anything unless you know the voltages that are involved. And when you start to change those relative voltages, you will drastically change the compensation percentage, even though the actual spillover hasn't changed. So keep that in mind. Yes, a very, very high compensation value is something that you should look at. It's a potential red flag. And you might be able to lower it by changing the relative voltages between those channels. But if it remains high, then you just have high spillover. So high compensation could mean very high spillover, but it could also simply mean imbalanced detector voltages. All right, how can you avoid data spread when using bright fluorochromes? Well, there isn't really a way to avoid it, but what you can do is you can choose things in the spillover channel that don't require high sensitivity, that aren't really, really dim. And so, if you have a PE Texas Red conjugate and a PE conjugate, you can use those together. That's fine. But either they should be mutually exclusive populations, as I already talked about. That's the best solution of all. Or the thing that you're looking at in PE should be sufficiently bright that the data spread from the PE Texas Red doesn't kill it. It still allows you to resolve the dim positives. And yes, I agree. When people look at things like CD25 high, which is a smear, that's very hard to do when you've got a lot of data spread in that channel. You really don't want to be in a situation where you have a lot of spillover to the channel where you're reading out CD25. So BDC-S&T. Again, we don't want to be dwelling on any particular manufacturer systems, but since the question is here about PMT voltages, it is a good place to start, but it's not correct for everything. So the idea of that system was to give you a starting point that might be applicable for work with PBMCs, let's say. But there are certainly cases where you might want to change your voltages a bit, either because things are going off scale or because you can maximize dim resolution by moving things up a little. So it's really just a starting point. And no, as I kind of alluded to earlier, I don't like to use isotype controls on a routine basis. And for the most part, if I use a control for gating, that is other than a biological control, it would probably be an FMO. Is there a difference in setting up compensation with beads or with cells? And no, you can use either. Usually beads are simpler and brighter than cells, and you don't have to waste your cell sample when you do them. And the only caveat is if you use beads for your single colors, then use beads as your unstained population and vice versa. Make sure they're matched. But you can do 3 colors with beads and 1 color with cells if you want to. That's okay, as long as you have a negative population that's matched. And your beads should be at least as bright as your cell population. Is it possible to run 3 to 5 colors without compensation? Sure. You'd have to pick exactly the right color combination, but you could. I'll bet you could do something like FITC, APC, and maybe one of the violet fluorochromes and have no compensation. Or you could probably even choose a set of Q dots or brilliant violet dyes that you could put together that have almost no compensation. So that is possible. But once you get up to 6 or 8 or more, there's no way. How important is a proper titration? It is important, and not just for good compensation, but for good resolution sensitivity. Oftentimes, the manufacturers have pre-titered things, and they're perfectly fine. Although you might find that when you do your own titration, you can go down a little bit and save a little antibody. But titration becomes particularly important when things are dim and used in multicolor combinations, and there you do want to pick the one that gives you the best resolution, the titer that gives you best resolution. All right, so yeah, I already answered the one about beads and cells. You can do some channels with beads for compensation controls and others with cells for compensation controls, but the autofluorescence between the two is not the same, so you need to match them. You need to have a negative population of cells for the ones you're doing with cells and a negative population of beads for the channels that you're compensating with beads, and that's the only caveat there. All right, we're pretty much at the top of the hour, so I think we should probably stop here, and I appreciate everybody's attention. Thanks very much for joining, and I hope it's been helpful. You can feel free to send any other questions my way, and we'll try to address them. Thanks very much.